Hi everyone, I am Dr. Anjugam, Assistant Professor, PG and Research Department of Microbiology, JJ College of Arts and Science, Pudukkottai. I am going to talk about sources of microorganisms in air. The content included in this topic. Before going to the topic, air is not a natural environment for microorganisms. Air is not a medium. It does not support the growth and reproduction of microorganisms. Air is not a carrier of microorganisms. The common microorganisms present in the air, that is fungal spores and the bacterial spores. The main sources of microorganisms are soil, water, plant and animal surfaces. Humans produce bioaerosols. The bioaerosols, that is droplets, droplet nuclei and infectious dust. And other sources of microorganisms in air is sewage treatment plant, food and paper processing plant, slaughterhouses, human activities like dusting, brooming and washing, etc. And then, what is aeromicrobiology? The study of intramural and extramural microorganisms in air. Air is not a natural environment for microorganisms. The physical and chemical parameters prevailing in air do not support their growth and reproduction. That is sunlight, UV radiation, desiccation, lack of nutrients, etc. Hence, the amount of microorganism in air is less than soil and water. And then, distribution of microorganisms in air. Troposphere, high load of microorganisms. Vertical distribution of microorganisms, that is air current, wind blow. What is meant by air current? Moving of air that flows in a specific direction, that is air current. Wind blow means differences between the pressures the air move from the high to low. The horizontal distribution, that is physical and chemical factors. Microorganisms considerably reduces as altitude increases. At higher altitudes, microorganisms are exposed to temperature, oxygen, atmospheric pressure, water availability and organic carbon. And then the main sources of microorganisms in air, that is soil, water, plant and animal surfaces, human beings, as I told in the previous slide. And then suspension of microorganisms in air. Microorganisms remain in the air for varying time periods depending upon speed of air currents, size of particles, humidity of the atmosphere. And then first we are going to discuss about main sources is soil. Soil, the most common source of microorganism in air. Liberation of microbes to air from soil by environmental activities like wind blow, air currents, and human activities like digging and plugging. An active soil environment liberates more digging microorganisms. Rich, fertile and cultivated soil shows a higher viable count than sandy and uncultivated soil. Soil covered with vegetation liberates less amount of microorganisms than bare surfaces which are easily acted upon by wind and air currents. And then water. Microbes liberated from water into air as droplet or aerosols. Environmental activities, wind and tidal action. Human activities is swimming and water sports, etc. And then plant and animal surfaces. Microorganisms on plant and animal surfaces can be commensals or pathogens. What is meant by commensals? Organisms without benefits from another organisms, that is without harming or benefiting it. Pathogens we know very well. And then 70% of plant diseases are transmitted through air, mainly fungi. Plant pathogens spread over long distance. Example, spores. Animal diseases are less frequently transmitted through air. Human beings. Human beings, the main sources of microorganisms in the air, that is bioaerosols, contain microflora of mouth and upper respiratory tract. Activities like coughing, sneezing, talking, laughing, singing, etc. liberate bioaerosols. Aerosols means particles suspended in air. Aerosols with pathogenic bacteria, viruses, microbial toxins, etc. and can cause infectious diseases on inhalation or ingestion. And then human beings continued. Bioaerosols vary in their size and composition. That is the size ranges between 0.02 to 100 milligram. Based on size, aerosols are divided into different kinds, that is droplet nuclei and infectious dust. Composition based on the type of microorganisms or toxins and also the type of particles like mucus and dust, etc. Then droplets formed by coughing, sneezing, talking, laughing, etc. and also during diagnosis like suctioning and bronchoscopy. Consist of saliva or mucus, epithelial cells, cells of the immune system and various microorganisms. Droplets, hundreds of microorganisms in droplets, and these organisms can be respiratory tract pathogens. Size of the droplet determines suspension period. And then usually a large size greater than 10 mm or more will settle rapidly in still air. They travel less than 1 meter through the air and are deposited on the nasal or oral mucosa of the new host or in their immediate environment. If inhaled, they are draped on the moist surfaces of the respiratory tract and cause the upper respiratory tract infections, will not cause lower respiratory tract infection. Next, droplet nuclei. Droplet nuclei originate from droplets by the evaporation. Droplets in a warm and dry atmosphere evaporate rapidly and the solid material left after drying up is called a droplet nuclei. Size less than 5 mm and contain microorganisms, dust particles, skin cells and other debris. And then droplet nuclei. The droplet nuclei widely dispersed by air currents, remain for hours or days, can escape the mechanical trap of the upper respiratory tract and enter the lungs. More potential agents of infection and important role in airborne transmission of diseases, particularly respiratory infections. The droplet nuclei in airborne transmission of diseases was first studied by Wells in 1955. Microorganisms present in droplet nuclei should be viable to cause an infection. The viability is determined by atmospheric conditions like humidity, sunlight, temperature, size of particles bearing the organisms, and the degree of susceptibility or resistance of microbial species to the new physical environment. And then infectious dust. Large droplets settle out rapidly onto various surfaces and include nostril and throat discharges of patients containing infectious pathogens. Remain suspended in air for longer periods of time. Liberates already told, sweeping of the floor, handling of contaminated kerchiefs. These are all the factors. A serious hazard in hospital areas. Laboratory practices like opening of frozen bacterial cultures or the cotton plugs tried after being wetted by the culture broth will liberate infectious dust. Next, thank you.